I'm doing a microbiology camp with UW, and it's at home, but right now we're on break, and earlier today I did my presentation about microbiology in the news. Ribosomes, where protein is made. An mRNA vaccine encodes proteins of a virus which, when inserted into a cell, should trigger the immune response to create antibodies. And then, so I got that over with, and this is like a month-long course. And then right now we're learning about mutations, and last week we learned about DNA and RNA. And then later today we're doing a lab, and it's with cabbage, so I'm kind of confused. But we'll see what that's like.
Microbiology camp had companies send me the material since we won't be able to do it in person. The first package I got was just 4 of these mason jars from Amazon, and they also sent a microscope in this big box. And I have 2 different lenses. Then from Fermentaholics, I have glass fermentation weights. There were 2 of them, but when we opened it, one of them was all shattered. So there's just one of these. And then from Home Science Tools, I got some microscope slides of algae, some methylcellulose, like glow germ stuff. Then the last thing I have is a protozoa culture kit, and there's some dry culture mix, a pipette, and a cup. And some things that they said they ordered for me didn't arrive, but I'm sure they'll sort it out.
For this lab, we made sauerkraut. We just cut up cabbage and measured that, and then put it in a bag and added salt. Then we shook the bag to get moisture out of it, and then we took it and put it in mason jars and then smashed it to get liquid so that we could take the pH.
My sauerkraut, and since one of my weights didn't arrive, I just used some pie weights, but it's the same weight. And then I'm just gonna let it sit, I guess, and I'm Zooming with some friends from the class. And I think we're supposed to hop back on the big class Zoom in 15 minutes.
This is my sauerkraut after a couple of weeks. The discolored one was on the counter for the whole time, and the brighter one was in the fridge.
Today we're doing some lab work, mostly just preparation for other days, but I have just a cup of water and then my protozoa culture mix, and I'm just supposed to pour it into the water.
Here's how my protists looked under a microscope once they had grown. Here I am looking at my algae slides under a microscope. This day we were learning about pathogens and symbiotic relationships.
Today I'm doing the glow germ experiment thing, and I've done this before in class, and you basically just squeeze a little bit of it onto your hands, then you rub it.
The glow germ kit shows where germs could be stuck on your hands and shows the power of hand washing. So here we're just in the dark shining it with a UV light.
Now I'm going to wash my hands with soap, and we'll see how much of the germs are gone.
As you can see, a lot of the glowing green stuff is off of my hands, which shows that I washed my hands well enough to get the fake germs off.
I was doing my quiz and for this week, and then we did all this, and then there was a question about horizontal gene transfer and how it was used in biotechnology. Sorry for all like the flashy things, but I did like my actual answer, but then I also put a story about me buying genes and giving them to someone, because I'm obviously the funniest person in this camp. So for those of you who thought that I didn't actually submit it, I did. Submission details, that, and blah, 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 blah. There's the question with my story Submitted. Hello, I just finished the second to last day of class. I have blue lipstick on, just, I don't know, for fun at the end of class. I did this once before, and my friend also used a blue lip gloss. It was funny because we both asked questions, and they didn't say anything. But anyway, I'm excited for the last day of class because this class it's been a month long, and it was pretty intense, I would say, because we had like a quiz every week and lots of experiments and just lots of work, and it's summer. It's the last day of class, and so I'm just doing this final presentation with some other people in the class and doing all these slides. And then after I present, my class will be over. Bacteriophages are pathogens that all target bacteria, such as E. coli, which is the bacteria that the T4 bacteriophage targets. Bacteriophages were first discovered in 1915 by Frederick Thwart and again in 1917